So today my presentation topic is about how I've used markers in breeding and molecular breeding, seed purity and in plant variety protection. I started out using markers to map QTLs for downy mildew in pearl millet, which led to the release of an improved HHB67 pearl millet variety. And then I moved on to using markers to map late blight resistance in tomato, which was a much more challenging situation, multiple QTL, very low heritability, and really was very intractable in terms of producing a product for breeding. So after that, I moved to industry and established marker systems in a SNP lab and started to implement them for seed purity. And in seed purity, we use the traveling salesman problem, which is a genetic algorithm to select the smallest marker set to be able to distinguish amongst all of our germplasm. And then we worked across industry. We worked with 5 different industry partners in the US and 10 industry partners in France, and established the first ever marker system to be able to be used in intellectual property in maize for global IP protection in maize. Since that time, I've started to use markers in genomic selection. And the most efficient way to do genomic selection is to be able to select very small, very efficient marker sets that can be delivered within 1 week of the breeder requesting them. So I've been working in a new area of markers called targeted sequencing. It has multiple names right now because it's very new. Targeted sequencing or next generation genotyping. And I've been developing those marker systems so they can be effectively used in double haploid breeding and other systems which have a very short turnaround time. During that time, I managed the sequence database and the SNP database and developed products that could be used to efficiently turn sequence into markers. And so now I am very happy to be working on the GOBII project where we're managing genotype data for breeding.